So, welcome everybody. I'm Tatiana Bazzichelli, curator of Transmediale Festival, and I'm really glad to announce another event of the stream Ashes to Ashes. And this night we are going to do another kind of journey, if we consider the event of yesterday, because we are going to reflect on the invisible data underneath our biological science and also our body. That is why the panel is called Under the Skin. So we are going also to investigate possible disclosures related to openness of data that are coming really from our organism and the way to combine art and science. And I'm going to introduce the moderator, Alessandro Delfanti. Actually, we know each other since a long time as part of the Italian activist movement, but then he flew away from Italy and now is working at the McGill University in Montreal as a postdoctoral fellow, and is especially working on participatory media and contemporary biomedicine, and also teaching an undergraduate seminar titled Online Cooperation in Daily Life. At the same time, he's also the author of a book that you can find in the bookshop. Just a bit of advertisement for your book. That's his name, Biohackers, The Politics of Open Science. So now I leave the word to you, and I welcome all the speakers on stage.
Thank you, Tatiana. And so yeah, my duty tonight is just to introduce the theme of this panel and our speakers, and then of course like try to start and help the growth of a conversation with you in the public. So we go under our skin tonight. So we stay digital, but we go inside our bodies, and inside our data coming from our body, from our bodies, and how those data can be repurposed, reinvented, and redistributed and reused in new and creative ways. I wanted to take a little step back and just talk a little bit about what's the condition of being digital today, according to me. First of all, I think there are two economies related to sharing, which is the issue at stake tonight. So the first one is sort of a political economy of sharing. So the data and the content we put online create the internet. Without our data, without our content, the content we produce, there wouldn't be any network, there wouldn't be any internet, there wouldn't be any service, there wouldn't be the social life that thrives on the internet, right? So there is, in a way, we are making it daily. We keep on making it by producing data and sharing them online. The second economy is a moral economy, so it's related to our values and how we produce sociality. We're more and more committed to that, to sharing more, to contribute more, to interact more, because that's how we build our social bonds. That's how we help our communities. That's how we make friends. That's how we maintain our relationships. That's how we create visibility for ourselves. That's how we participate in producing a society and our communities. So these two dimensions sort of collapse in the theme that we're going to explore tonight, because those data, those amazing data, if you want, that produce both our sociality and the internet, get burned and become ashes, as in Tatiana's metaphor, and get privatized and obscured, get buried in secret databases, as you all know, run by the government or by secret institutions, by corporations, that bury them under layers and layers of passwords, non-disclosure agreements, even concrete vaults, firewalls, and we cannot access them, we cannot use them, and they're used against us. So tonight we're going to talk about those data that are buried, but that can be resurrected in a way. So those ashes can become sparkles again and illuminate for a moment open possibilities that are out there, that are maybe unpredicted or unpredictable, but that can be interesting to see what our future could be if we could unveil and disclose what happened in those databases. Of course, you have to hack into them in order to explore those data, in order to illuminate them again. And of course, those sparkles can quickly become ashes again. So, and left us without nothing in our hands. But that's what we are going to explore with our friends here, which I'm going to introduce very quickly. So we're going to. My plan is to go deeper under our skins as we go with our panelists. So the first one is going to be Rüdiger Trojok. Sorry. He is a biologist by training, and is part of the collective Hackteria, which is a collective that does biohacking, so hacks into life, and he'll explain us how. He also works as a consultant for the German Parliament, working on openness in life sciences, so how open source approaches can be used in biology and in science in general. The second speaker is going to be Salvatore Iaconesi from Italy. A friend of mine since not since a long time, but now a good friend, I want to say. And he's going to go again a little bit while Rüdiger is going to be sort of general regarding the general evolution of open science. Salvatore is going to talk about something very personal, so his own data, his own medical data, and you'll know about this. So he's going to present a project, like a huge project, I want to say, that built an amazingly, incredibly huge public and community around this data. So you'll listen to him and he'll explain that. And the last speaker is Pinar Yoldas, Turkey, but now living in the US and working at Duke University. And she's an artist, and with her sculpture, she works in an art and neuroscience project at the Brain Institute at Duke. Is that correct?
Yes.
And with her sculpture related to data, she basically explores unexpected ways in which data coming from. Well, if in Salvatore's case it's going to be brain, in Pinar's case it's going to be emotions. So it's going to be deeper into our data. So we have a biologist, a designer, and an artist. And it's going to be very fun and very interesting, according to me. So I'll leave it to them, and then I'll start the conversation with you and I'll wrap it up at the end. So Rüdiger, you go first.
Yeah, thanks. Let's see if this is working. Mm-hmm. Yeah. One second, I need to fix the connection. Oh, there it is, okay. Where is it going? Yeah, this is just my wallpaper. Here we go. Yeah, I want to talk about biohacking. Maybe you've heard about the term. I'm a biologist by training, and I'm active in the Hackteria network, which is a collective of artists, engineers, scientists, and amateurs, laymen, who are interested in biology and the technologies and data and implications of it. So I want to start with a bit further reaching out. Komm schon. Around the 14th, 15th century, people started to look into space and got curious what the heck is out there. And we're looking at the stars, and then had the discovery that we are not the center of the universe, and the Earth is not flat. A couple of centuries later, in the 17th, 18th century, people looked into microscopes and saw that there are cells. And in the last century, people started to look even deeper into it, and what they found were something that kind of looks like stars, but it's atoms. And what I display here is actually a protein, which is also very much connected to the topic of brain generation. It's called the neural growth factor, and it's a protein that is crucial for embryonal brain development, as well as it's found after bungee jumps in elevated levels in the blood. It can be used against depressions. And what I also found really interesting, it was rediscovered in another context, that is sex. It is present in very high concentrations in sperm, as well as in the fluid that surrounds the female egg. And when the sperm approaches the egg, this protein is the guidance. The concentration gradient of this protein is guiding the sperm so that it can find the egg. And this is one of the many examples. There's around 100,000 of these protein structures freely available online. And this is one example that I stumbled upon in my studies and that I wanted to present to you as an extremely fascinating result that can be that was brought to light in the last 20, 30 years of biological research. So I need to torture you a little bit with the scientific knowledge about the biology, namely the transcription and translation. So how do you get from the DNA that is consisting of only 4 bases aligned in strings, packed in your cells, to the proteins, like the neural growth factor that I've shown you. It's an abstract code which is encoded in this picture in the middle. So it's always 3 of these 4 bases in combination that code for 1 of 20 amino acids. And you read it along the string, so always triplets determine 1 amino acid. And then these amino acids are again becoming a string, but this string is not one-dimensional, it's 3-dimensional. And it's folding in space, and it's dynamic, it's reacting in time and space, and it has certain properties. So maybe you can, it's like a sculpting mass, and you can do everything with it. So the whole biosphere is operated by these tools that are encoded in the DNA. So you can consider this system as something abstract. The information of the DNA is abstract, but it is physical in the same time. So I think the idea of Descartes from that we have a separation between the abstract and the physical is it doesn't work on this. It's the same. It's both in the same time. And based on this, the whole biosphere is based on this, and it's mainly separated, or you can mainly distinguish 2 different kinds of cells, which is the prokaryotes and the eukaryotes. The prokaryotes are really small, and they are rather old, like almost as old as the planet, 3.7 billion years, 3.6 billion years. It's not exactly clear, but really, really old. So basically the Earth cooled down, a lot of meteorites hit the surface, and then there was like carbon atmosphere and like a horrible landscape. We wouldn't survive it for a minute. And already in this stage, when the first oceans appeared, life appeared. The eukaryotes, which is us, we are eukaryotes, plants, fungi, insects, animals, all these higher animals that are mostly multicellular, organized in big patches of cells. They came about 1 billion years later, and they are more complex. And we are consisting of 10 to approximately 100 trillion of such cells packed together. And as a major distinction is how the DNA is arranged. So the DNA is much more complex than the eukaryotes, so it's packed in a special compartment called the nucleus. While the DNA in the prokaryotes is simple structure and floating freely in the cell. this is just like basic information that is crucial to understand, and also how to know how to use the data and how to manipulate it, because it's the three-dimensional physical representation of this abstract information encoded in the DNA. So there's these two different types of appearances, which are still connected because they all life forms on the planet use the same code, the same mechanism of reading the code. So in the end, we are all relatives, all living creatures on this planet, from the most stupid cell in, I don't know, deep sea to us. We are kind of like the same. There's not much difference between us. So the hackteria network is, as I said before, an international collaboration of scientists. And I want to also use the chance to pitch this meeting in April in Indonesia. So if you feel like, you can fly over and join us. And there's a 2 weeks hackathon session where we come together and build stuff and talk about the implications of these insights that the research in the last 20, 30 years revealed. The events that are hosted are very diverse. So we had some like small-scale hack sessions in Switzerland on the top left corner. There was a lecture and workshop series in Medical Museum in Copenhagen. There is some other people which are remotely connected to us, doing like street education of genetics in Ankara on street festivals. And this is a picture from a hackerspace, or I think it's a basement of someone in Indonesia, where the people started to build generic lab equipment. So that is one of the main things, is to build cheap and available lab equipment so that you can use the technology that is usually very expensive but really not sophisticated from the institutions, and get it in your own hands. Hackerspaces can look really diverse. So you need, of course, you need technology, you need tools to access that knowledge, but it's not that unimaginably complex and inaccessible. So the most simple setup is this one in the left corner. It's a pressure cooker, a stove, and ingredients for soup. So everyone who made a soup and it happened to start smell bad did this kind of biohacking experiment, because you just grow bacteria in a soup. They really like it. I mean, we like it, we live from it, so the bacteria is not so much different. They like it as well, and they just eat it and proliferate. Then there's other places, like in Paris, in this garage, where people got together and collected some equipment, old equipment from universities, for example, put it together in this and set up their own lab. This one is a picture of the flat that I had a couple of years ago in Freiburg, where I set up my private lab at home. And this one is the most sophisticated and more structured space in California, which is in Germany you would call this a Volkshochschule, I'd say. It's like a public place where you pay a little and you get education. But the common point is that you don't have to be enrolled in a program in university, or you don't have to have a title to be able to use it. So it's open to anyone who wants it. I want to introduce you to some projects that were done. This machine that was built by a Swiss engineer, Urs Gaudenz, is used to copy and paste DNA. And what it does, it is heating and cooling. That's basically the only job it does. You can program a script on the computer, you connect it to the machine, and then the machine controls a small aluminium block, and that heats and cools down to very precise temperatures. So you can say, okay, go from 95 degrees to 72, and then again up again. And this temperature control is needed to control enzymes, which you use to copy and paste DNA. So you can, it's not as easy as on the computer, where you just push some buttons, it takes some more time, and it's not so precise, but basically you can operate it as an abstract code, use it as an abstract code. You can also order it from companies. That's a bit more expensive but easier, so you just write a computer script with these 4 letters, A, C, T, G, in various combinations as you want it to produce a certain gene. And then you send it to a company and they ship it to you, and then you can do the next steps with it. like reprogramming cells. This was an approach I did with an air pressure device, which I built myself. In the end, the model that is displayed here, this kind of stick with the air pressure on it and a trigger, did not really work. So I went to even a more simple setup, which is a whip dispenser. You know, it's like you put the gas in and then the cream, and then you just like get the whip out. So it has like 40 bars pressure, and it's enough to accelerate gold particles, tiny, tiny gold particles, like 10 micrometer small gold particles. And on this gold particle, you glue the DNA that you ordered or prepared before. And then you put these gold particles in the tip of a pen, and you connect it to the whip dispenser, and then you just shoot it on. Here I used an onion cell. And the code that I programmed was a small protein that is fluorescent blue, and it is located in the nucleus, the structure that contains the DNA in the cells. And you see here in the bottom there is this brighter patches. This is the nucleus, and there the DNA code is where the DNA code is stored, and the protein was directed that was coded in the DNA that I put in the cell was directed to this nucleus to color it, to highlight it. Another experiment was baking bread with reprogrammed yeast. So this picture I didn't make myself, I just got it from Wikipedia. It's an electron microphotograph of a yeast cell. So I took such yeast cells, normal baker's yeast that you get in the supermarket, open up the cell, take out some DNA, open up another cell and put it back in. So I just shuffled yeast genomes around, basically. So it didn't really do any specific task, but I did all the steps that are necessary to manipulate a cell. And then I baked the bread and I ate it myself. The Gesundheitsamt required me that I only eat it myself because the regulations are really tight in the EU. I was not allowed to give it, not even let others touch it. So it had to be contained and I had to eat it myself. I didn't suffer anything from it. There's another project that I found really cool. It's controlling cells or small micrometer-sized structures, like this bead that you see in the middle there. There is this red mark. Next to it, there is a small bead. It's a plastic bead of 15 micrometer size. It's a photo of a microscopic picture. The microscope here can be seen in the upper corner of the lower picture. It's a webcam. So we turned around the lens of the webcam. So it's magnifying, about like 400-fold magnification. And under it here, there is a sample where the image is taken from, what you see on the upper side. And under this sample glass slide, there is the inside of a DVD burner. So we took out the DVD burner and built a control to control the laser of the DVD burner and focus it on this particle. So we use, and the red bar that you see in the picture is the reflection of this laser. And we managed to focus it on the bead, and then you can trap the bead, you grab it, and you can move it around. Just with a light. It's like a tractor beam in Star Trek. It's just like a small thing approaches the spacecraft, and then the tractor beam grabs it and then just moves it around. It works. So, talking about data, just last week there was this report in the Spiegel that now you can get your genome for €1,000. And this is kind of incredible. So the costs for sequencing a genome dropped dramatically. So you see the time scale here in the last 10 years, and the green marks are the actual costs for genomes, and this purple mark, the purple line, is the Moore's prediction. You know the Moore's law from the computer chip technology is so that the price halvens every 18 months. So the sequencing is even faster. It's dropping dramatically. This leads us to the next consideration. we sequence genomes with this, we sequence species with this, like reading out the DNA information of species. And so far we have 4,000 species sequenced, and these are the known sequenced genomes per year until 2012. Now we have around 4,000 sequenced genomes, but if we go on with the same speed, and this is the Moore's law, so it's even the conservative prediction. If we go on with this speed, we will have 1 million sequenced genomes by 2030. So now 4,000, and 2030 will lead us to 1 million. And it's not so far away. So what the heck are we going to do with all that data? And please don't do this. Please don't avert your eyes from this development. I know it's outrageous, it's crazy, it's amazing. But please don't leave the scientists alone with this data. It's a cultural phenomenon. I think everyone has to interact with it. It's life itself. So my suggestion is that we treat nature as a common good, because everyone has access to it anyway. Have all this information freely available, and also have the technology to access that information available, because if we don't have the access to it, it's still a monopoly. We cannot use it, and then it's still hidden. It's like a firewall, a physical firewall. And then I think in the future we will all be able to understand this data, involve in it, involve in the process of generating it and using it, and also create something out of it. Whatever it is, it's up to you. We also suggested a code of conduct first, because we have to be careful how we deal with it, so we have to reflect on that. Maybe it needs more discussion. I invite you to get into this. This is the biohackers that have put their marks on the Google map. Now it's 2 years old, actually. I think there's more now. And this is all the hacker spaces on the planet. So I hope that we get all these hacker spaces equipped with the technology to also hack biology, so that we have even better access to it everywhere. Thanks for your attention. I'm done.
Thank you, Rüdiger. And from this dream of a more collective, more distributed science, we're gonna go through again to a very personal, if you want, project, which is Salvatore's, but that still gather a collective response. So, Salvatore, what's the data you are hacked into?
Some very personal open data. So this was, luckily the keyword here is was. This was my cancer. You can see that the little bright spot all over the images. These are several slices obtained through an MRI scan of my head a few years ago. Last year, it seems more. So I had the brain cancer. I had the brain cancer. It just appears. It appears in many people's, in my case, heads. It appears all over the body, in people's bodies more frequently each day as we go by. The statistics are 1 on 3. The current statistics are 1 on 3 people will have to deal with something which is called cancer, tumor. And so I had mine. Okay, we don't want to miss anything in life. I had my cancer. So what goes on? What happens? What is the first thing that happens when you have cancer? The first thing that happens is that you disappear. You, as a human being, disappear. People start talking about your cancer instead of talking about you. And that is incredible. People start, you could actually not be there at all. You become a set of data. You become a set of data, blood pressure, exams, images. You become a set of data. You don't actually need to be there. The doctor, when the doctor visits you, he doesn't look at you. He looks at the data, the images. You can go away. You can leave the hospital. You can do whatever you want. You actually disappear. And this is incredible. This is incredible because it's an incredible situation. You disappear for doctors, you disappear from your friends and family. When people say hello and you have cancer, they don't say hello to you. They say, how's it going? What do the doctor say? How is your pressure? How are your images? They talk about your data. Okay? They don't talk about you at all. And this is very incredible. While I was in the hospital, I started realizing this thing in a very intimate way. One day, when I was in the hospital, I asked for an image of my own cancer. So I wanted a printout of the image of my own cancer because I wanted to talk to it. I wanted to look at it. I want to speak with it. I want to find agreements with it. Okay, since you're there, do something useful. Okay, no, give me some superpower. Well, who knows what? Okay, be useful. Okay. But basically, I wanted to look at it because nobody wants to look at the picture of his own cancer. And so I asked the nurse, so is it possible to obtain an image of my own cancer? No, it was not possible. Because there were many problems, legal problems, insurance-related problems. Imagine a hospital. Okay, what happens if we told him that he has a cancer and it turns out that he was not a cancer at all? Will he sue us? So legal problems. Privacy problems, regardless of the fact that it was my own cancer, there were privacy problems. You have to ask the permission of blah, 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 bureaucracy, administration, etc. So in the end, to make a long story short, I was not able to obtain an image of my own cancer. And so this started sounding really strange to me, okay? So that's when I decided to leave the hospital. Because this situation was really weird. Imagine it. Imagine, okay, people not talking about me, not talking to me, not talking with me, okay, but talking about my data. And this data, on top of that, this data is not even accessible to me. Because, okay, I cannot even obtain an image of my own cancer. So what did I do? I left the hospital. Before leaving the hospital, I asked for my medical records in digital format. So I went home, and I was very happy because I thought that I was finally able, I was finally going to be able to obtain my image of my cancer. So I stick it came under a CD. I stick the CD in the reader, and what happens? It comes under a DCOM format, D-I-C-O-M. DCOM format is, technically speaking, is an open format, but with a twist. When you look at images on your email, on social networks, on all the things you do every day, okay, you manage, you have learned to manage PNG images, JPEG images, the more esoteric of you will have used TIFF images, okay, but no DCOM. Where's DCOM? Where's DCOM? How many of you have used DCOM? Okay, you cheated because I use them. Okay, not many of us know how to use DCOM. And so, what is DCOM? DCOM is, again, an open format which is meant to be used by professionals. So, the standard person, when I do this thing, I measure up with my dad, okay? What would my dad do to open a DCOM file? He couldn't. He would have to find a certain software. Download this certain software, install this certain software, open it up, and see that this software uses a language, no linguistic, in linguistic terms, a language to which is not used at all. Okay, the terms are different. It's not images. It's MR blah 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 blah, which are not used at all. Okay, as a human being. So, because it is simple, because it is a format which is meant to be used by professionals. Okay, so I was given my medical data in a technically open format, technically speaking, in an open format, but to take this CD and give it to another professional. Again, my data, as long as it was, even if it was open, this beautiful word, open, accessible data, it was not meant for me. It was not meant for me, because the regular person does not have access, it does not know how to use this data in an open format. It's incredible, okay? So I was not able initially to have my image of my own cancer. It was a nightmare, okay? I was really frustrated. So I went and searched, I know how to use computers, blah, 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 all that and that, and I was finally able to open up this decom file and print out my image. But I thought that this was the situation in which millions of people, we're talking about 1 on 3 people all over the planet have to deal with. So this thing really, really moved me. This thing of this legend of openness and this legend of data access, etc., etc., really struck me because there was one main point. There was one main point. The main point was that the data was simplifying the human being. The whole system was describing, was dealing not with Salvatore Iaconesi, but with a very simplified version of Salvatore Iaconesi, who had lost all its complexity, all his social connection, all his relationships, all his everything, apart, and kept only the data, and this data on top of that was not accessible to the human being Salvatore Iaconesi. It was a nightmare if you think about it, okay? And this is how medicine works, this is how science works, this is how things work. It was incredible. So what did I do? I did this. I turned all my images, records, etc., in formats that were accessible for human beings, not for patients, not for professionals, not okay. And I put them online. I put them online, so in JPEG, PNGs, movies, so because the files, the image files have also animations. And I put up this website, which its name is La Cura, and I made a video message telling the desire to really open up the process, to really open up the process, but to whom? To really open up the process to society. So to reclaim, by publishing this data, I use this data as a metaphor of the condition of the patient that was a simplified version of the human being. So I wanted to become a human being again, not a patient, not a set of data that was handled by professionals, and I reclaimed my human beingness. Okay, I reclaimed my fact that there was a human being. So I brought this data back into society because I wanted to be back in society. And so in the video message, I asked for the collaboration of potentially all members of society, be them persons, artists, designers, whomever, even researchers, to provide me with a cure. Okay, so it's clear how a researcher or how a doctor can try to offer me a cure. But how can a designer offer me a cure? How can an artist offer me a cure for cancer? How, for example, they can speak to each other to communicate, to provide a better cure. So all these things. So I brought back the disease into society. You know? For those of you who remember Foucault or Basaglia, no? The whole problem is that disease no, is encoded and is out of society. So how can everyone help? And so the cures started happening, started arriving. So there millions of cures started arriving, like up to now, because this is going on, like 900,000 cures arrived up to now, so lots of cures. Artists try to provide their form of artistic cures. This is Francesca Fini with the performance she designed for La Cura. Or, for example, this is Patrick Lichty. Patrick Lichty used the data, the images from the scans from my cancer, and used them to produce a 3D model of my cancer and put it on Thingiverse. So now you can buy it and you can have my cancer too, if you want. Okay. So no need to be 1 on 3. You can just purchase it, okay? So you can save yourself. And this is like just like a small part of all the cures. So this is a zoom in and this is a zoom out, but it's also much bigger. On the columns there are the topics, so if a person is talking about how to use marijuana or LSD to cure cancer, there will be a column for that. And if the person mentioning marijuana and eating habits, there will be a line connecting to two columns, okay? So many cures started arriving. And it was beautiful because, as you can see from the intensity of the connections, it was like being in a state of constant translation from one domain to the others, because it turns out that there are many weird cures for cancer, supposed cures for cancer, very even dangerous cures for cancer, even weird cures for cancer. According to a person, I should take a trip to Argentina because it will cure me from cancer completely. So go to Argentina if you have cancer. So many ways. So, but in other ways, many things are just translation from one language to the other, from one domain to the other. There are many things that take their birth, for example, from traditional medicines and find their application in philosophy and in science and in many things. Okay, so many. If you look for example, on the website at these and then these connections, you will find many interesting things. But what yes, I'm finishing. So what's the purpose? To become a human being again. Because when we speak about data, we are like really running a danger, the danger of forgetting human beings and society and the data and the fact that the data is really talking about them, but it's also a very simplified version of people and what they do and how they live their lives. And for example, this turns out that it has been a wonderful way to bring health back into society, for example. So in society we are used to taking into consideration healthcare from as something you buy. So you have the insurance, you have the things. So healthcare normally is something you buy, either through taxes or through insurances or something. And we moved instead to a notion of health, which is something you do instead, together with all society, which is a very interesting concept. And most of all, it is turned into this incredible idea, which is more than an idea, because it actually took place of an emergent peer-to-peer welfare. So the idea and practice according to which health and well-being in a society is something that can happen only if all members of society have well-being, and so the whole of society takes care of their peers. Okay, and I want to leave you with something, with just a little something. If the video starts, so this is a short video, and it. Every dot is a person, and every connection is a connection that formed between between people in La Cura. Let's play it again. So it's just a few seconds. So persons and relations. Okay. And what struck me was that first of all, I don't know where I am in this. So to get the measure of how the many people and how they got involved. So I don't know which dot I am. I'm not central to La Cura. And the most striking thing of all is that if you look at it, this thing grows like a cancer. And so for me, this was an amazing discovery, because from my little point of view, I figured that this kind of process can transform an awful thing as cancer into a form of life, through society and through aiming to gain our humanity back, going beyond data, okay, and seeing that and bringing back the complexity of human beings again. So even a thing as awful as cancer can become a thing of life, okay, and that's all. Thank you.
Thank you, Salvatore. And well, my favorite cure is one that says to mix together aloe vera, honey and whiskey, but it's not clear if you have to drink it or to put it on your head. So what did you do with that? Did you drink it or did you just put it on your head?
Do it, I like
Oh, okay. Anyways, thanks, Salvatore. And I think it's this problem of data oversimplifying reality is a good introduction to Pinar, because of course art can turn these data that are meant for a strictly scientific or medical use into something that unveils different dimensions, right? So I think it was a good introduction to you, Pinar. So let's go to your brains.
Okay, my brains. First off, I really want to thank you, because I'm so impressed and moved by this entire story. Yeah, and presenting after you is a big challenge, because now, I mean, what I'm doing seems so trivial and stupid, but let's see. I am an artist, trained as an architect, but currently, I don't know why I embarked on this program. I'm doing a PhD, learning neuroscience. I fell in love with brain, so I decided to study it together with my art practice, which wasn't a great idea in terms of my sleep schedule, but anyways. So I'll first, start my talk by giving you some examples or news from the field of neuroscience, which studies the brain, obviously, and then I'll try to collect it to big data. And then to wrap up, I'll show you, I think, 3 projects that are most directly related to brain, because I have many other projects that are not, directly linked. Of course, everything is processed in the brain, but anyways. So 3 developments in neuroscience. The first one is connectome, or connectomics. It's basically mapping the wiring diagram of the brain. This project have found, or this kind of field have found incredible support, financial support, especially in the United States. Just like the field of neuroscience, In fact, neuroscience is kind of the rising star of science in the States lately, kind of following in the footsteps of a Human Genome Project, not really reached that kind of budget yet, but the government invests in it. Obama, on April 2nd, 2013, like signed up this kind of, what would it be, like budget thing, which made most of my neuroscientist friends super happy. They're tech moguls, people who come from the world of media and technology that have been investing in the brains, such as Paul Allen, who is the other half of Microsoft. He has this institute called Allen Institute of the Brain. Or Jeff Hawkins, who wrote this pretty awesome book called On Intelligence. And he's like the inventor of Palm. Do you remember that Palm? There was that thing. Now it's all forgotten, but it led to other developments, of course. And he's leading the Redwood Institute of Neuroscience, trying to build truly intelligent machines. So there's, I would say, private and public support and funding for neuroscience. And connectomics is, I'll just play a video if I can. I think this is like the retina, the visual cortex of a mouse. But they were able to map the entire wiring diagram of C. elegans, the swarm. And now the goal is to map the human brain. Basically, what they're trying to do, what the science is trying to do is to map every single connection that's happening between every single neuron in your brain. And we have so many neurons, 85 billion at the last count, say. Before it was 100 million, it dropped down to 85. Maybe it'll drop down more in time, we don't know. But so this is a project, and this is actually really interesting because there's a Turkish poet, and he asks this question, and I'm going to translate it into English, and it will totally lose its poetic meaning, but he asks, can you paint happiness? Can you draw happiness? And in fact, with the Connectome project, you can see every single connection, let's say, at a moment where you're absolutely happy. So, but brain science in general is a science that heavily relies on data. So we look at the brains, we think we see the brains, but what we're looking at is basically visualization of, let's say, the blood response, like whether your neurons need blood, because that's the basis for fMRI, and it'll tell you what parts of your brain is getting activated, or EEG, or lately there's this new technique, TMS, which is the opposite. It basically doesn't picture the brain, but it stimulates the brain, etc., etc. So there are more, yeah, there are really beautiful sculptural or 3D models of the connectome as well. And there's this kind of interesting guy, kind of a performer artist, Sebastian Sung, who wrote a book called Connectome. So it's kind of hot lately, this topic. So yeah, the other new development or kind of interesting development is tracking thoughts, basically real-time fMRI neurofeedback. And I was affiliated with the group at Duke University, Alison Adcock and her lab members who have been working on this project. And I don't know how familiar you're with fMRI, but it's basically, you know, scanning your brain to see which part is getting activated. And it has amazing spatial resolution, but it doesn't really have that much of a temporal resolution, which means you won't be seeing things as they happen. There's usually a delay of 6 minutes or less. But with this new fMRI feedback technology, we can view the brain as thought happens. So this is kind of a very exciting development, and I know so many people getting, you know, psyched about this, and yeah. But the problem yet is fMRI machines are huge. It's this really loud tunnel that you have to lie down, so it's not really possible to like track thoughts outside the lab settings. And the last one is tracking emotions, and this has become pretty widespread. There are many ways of doing this. You know, we all know the whole lie detector story, but lately there have been a facial expression analysis software going on, and it's also become, I would say, corporate, because there are at least 2 companies in the States that have been selling this software. So I'm just going to play, and they also have like really funny names. Emotiant is one of them. The other one is Affectiva, very creative. So let's play. And the whole promise is that, okay, I don't want to dive too much into detail, but there's this guy, his name is Paul Ekman, and his like great influence on affective neuroscience, because based on, I would say, Darwin's theory of like emotional expression, which is universal among humans and primates, etc., we can detect the face muscles and, you know, certain expressions to kind of find out what emotion triggers that emotion. And these technologies are based on that. If you have seen Ekman faces, you'll recognize in this video that the humans, you know, actors playing aren't that natural. They're like really doing Ekman faces. They're like, because Ekman goes like this and you're angry, and he goes like this and you're sad. So they're kind of doing that, which isn't very realistic. There he is. But so this is a technology, and Affectiva has a very, you know, successful software too. And there's another device that Affectiva released, but I can't see it on their website anymore. And Affectiva is linked to MIT. It's a product of Affective Computation Group. They made this bracelet that would detect your galvanic skin response, measure your heart rate and your body temperature, to stream information about your arousal levels. So arousal levels are very important in affective tracking or emotion detection, because if we're not aroused, we usually are in a lame state of mind and we're not like really feeling anything. So that's like the first step. And so far, there's galvanic skin response, which tracks whether your skin gets more moisty, like, because, you know, sweat glands respond automatically. We can't really control it. Gaze tracking, face tracking, I've shown examples. Heart rate, again, hormonal, we can't really control it. Some, I think, meditation gurus can do. And bodily fluid samples, blood, urine, and even tears. Yeah, I recently read this paper where they were talking about how emotional tears that are, you know, shed after like an emotionally moving event have more hormonal content than just physical tears that are released to protect your eye. So these are all ways to, you know, track what's going on in our heads. And so on that sweet note, I just want to kind of wrap up this part of my talk with a personal experience. I was invited to a health conference in Palo Alto last year. I never understood why they invited me, but anyways, I went, and this was all these high-profile executives from insurance companies, some, you know, not CEOs, but hospital directors, etc. And they were discussing the future of health. And all they talked about was big data. And we were talking about these tracking methods and how they could be applied to, you know, in their, you know, future policies. And I heard things like a smart shopping cart, and when you hold it, it could, you know, track your heart rate, and it could tell where you're stopping, how long you're stopping. And the shopping cart is also kind of a spy. It'll tell the insurance company what you're buying. So if you're buying lots of vodka, I don't know why I said vodka, but in the context of German beer, or I don't know, like junk food, you know, sugar, fat, then you get like low points on your insurance profile. And that just blew my mind. I was like, oh my God. And they were also talking about shoes, running shoes, or like watches, etc., that would track every single thing that you do during the day, which would be kind of streamed to the insurance company. And I was like, okay. All right, on that sweet note, yes, I'm personally interested in the brain. My personal interest actually started with philosophy because I was really pissed with this whole like mind-body dichotomy, and I read this book called Descartes' Error by Antonio Damasio, and started taking neuroscience classes afterwards, because as an artist, I always felt like emotion was quite important in my work, and speaking to emotions or activating emotions. And unfortunately, emotions are kind of looked down on. The brain is thought to be the seat of intelligence and reason, but we don't really think about the brain as like the seat of emotion as well. And the study of emotion really kind of got me going. And this project that I'm going to talk about, Limbic, has a lot to do with that period of my life and my relationship with Descartes, etc. So in this project, I work with a neuroscientist, David Paulson, and we basically get an it was very easy for him to get an fMRI scan of his brain. It's usually quite expensive. It's around $500, and you have to go through procedures if you're healthy and want to get one. And after that, we sat down together and we mapped down this part of the brain called the limbic system. So it's named by Paul Broca, Le Grand Lobe Limbic, which used to be thought, I mean, for many, many years, neuroscientists thought this was where emotional processing happened. Now, lately, there are theories that claim that, emotional processing is more distributed than like just, sort of organs or parts. But I was still really thrilled by the idea that there are these structures deep inside the brain. They're called subcortical structures. They look beautiful because when you think of the brain, you just think of this like squishy, wrinkly thing. But deep inside there are all these architectural, sculptural forms which process emotion. Moreover, the limbic system is shared across mammals. So it's not only a human thing, it's something that we share with many other non-human life forms, which actually connects us. It's basically why you can, kind of have a sort of dialogue with your cat or your dog. And it's also the reason why your fish will never get so close to you as your cat. So well, these aren't David's brain, actually. I lost David's scans, so I'm just using generic imagery. But what we did was we got scans of his brain. We had like 64 slices or so, and then we mapped parts of the limbic system, which is, the core of emotion. And then I sat down and traced all of them manually and sent it to a laser cutter, and we got this sculptural form. Here you can see, and we color-coded different parts. So this was kind of a visualization of David's emotions as well. And you can see like the putamen, I think part of the thalamus, not sure. And then we hung these pieces in the brain space where they would normally be, and we got the sculptural form. We like picked 6 parts that belong to the limbic system or that make up the limbic system, and this was the result. So that was limbic. The next project I'm going to talk about is a little bit different. And I only have 3 minutes left. Awesome. So thinking about the brains and like brain development, etc. I was thinking about this notion, designer babies, and there are companies in New York which will, promise you that you can pick the eye color of your offspring or the skin color of your offspring. And since, brain development is also controlled by genes, I was like, okay, that's kind of superficial to play with the eye color. Let's play with the brains. And this is a snapshot from IBM's I Did a Baby, where this is the future, guys. Like every single thing the baby does, Chris, will be measured, and everything will be streamed. So I was also thinking about this iBaby, and I designed babies, which was fun. And the first one is high cortex, and PFC, prefrontal cortex, is where high-level emotional processing, language processing, and decision-making happens. It's also thought to be the place where thinking about future, planning the future happens. It's the part of the brain that's thought to be uniquely human. I don't know about that. That's what some neuroscientists say. But it's also, it's the executive center of the brain. And I was in Turkey last summer just watching how our president sucked. He basically lacked language skills. He lacked the imagination to, predict the future. He lacked social and emotional decision-making skills. He was a total prefrontal cortex disaster. So I said, okay, apparently this guy who's, has this kind of primitive brain cannot respond to the highly complex need of a young 21st century nation. So let's design a baby which would parallel process the needs of more than 70 million people uprising in the streets. So that's high cortex. This is how high cortex looks like. BA25 was more focused on emotional processing. Again, I was inspired by the chaos in Istanbul. This time, the city itself, after getting stuck in traffic for 2 hours or totally losing it, I was like, I can meditate as much as I want, but I won't be able to, maintain a good emotional makeup here. This city drives me nuts. And BA25 is Broadman Area 25, and it's thought to be the part of the brain which controls your emotional response under high levels of stress. And if you're a severe depression patient, you get zapped here as well. So I played with this area in BA25 baby, and I created, the goal was to create this baby which had this Buddha nature, loving, accepting, strong, poetically engineered, who can survive even in total chaos like Istanbul. So this is BA25, peacefully resting. And the last one is kind of a cliche, but it's humanimal. And this time I was thinking about these, really, really impressive human beings. Oscar Pistorius, who was back then my hero, then he shot his girlfriend, so he's not my hero anymore, but I still like Amy Mullins. The human nervous system is really capable of, expanding its space, and there are like brain maps that would evolve. Let's say even if you wear a hat, your body would know that your body border is expanded. So we have this amazing capability to adapt to extensions or expansions of the body. So in the humanimal, which you see here, the skeleton remains the same, the brain, everything else remains the same, but we have animal muscles. So it's a baby with animal muscles which can run and jump and do amazing things. I think I almost ran out of time, but maybe I can show this last project. And this project is the total opposite of the previous ones, because this is basically an organism which doesn't have a brain. I took out the brain from this organism. It's full, a vastly diminished animal. It's just made up of circulatory, reproductive, and integumentary systems. And it's basically a chicken or a poultry that doesn't have the unnecessary parts. So here's full swole, and I was looking at images from the corporate food industry in America, and this notion of cage-free chicken. I mean, chicken used to be cage-free. Who put them in cages, first off? And there were all these problems with putting the chicken in the cage, such as giving it hormones so that it overgrows, can't walk, falls down. So there's this problem with space. When the animal is caged, it basically can't move. And there's this amazing theory about the birth of brains, where they claim that we evolved brains only because we needed to move. There's an example for this. It's a sea slug. The sea slug starts its life as a happy moving animal. And it swims around and looks for the perfect spot to, continue the rest of his life or her life, finds the spot, settles in. The first thing it does, it eats its own brain. Because from that point on, it doesn't really need to move. And it continues its life as a plant. And again, in American academia, this is given as an example for tenure track, where once you get the job, you don't really need your brain anymore. But so I was thinking about that. I was thinking about movement and the lack of movement that these animals had to, deal with. And this is a regular happy chicken with all the biological systems it needs to move and to survive. And a regular chicken on the right. And my version, once you don't have feet, once you don't need to move, you don't need a nervous system. And once you don't have a nervous system, you don't need sensory organs. You don't need feet either. So my full swole is pretty diminished in that sense. And you can feed it with any nutrient input. And you can basically kind of design or modify the egg you want to eat. You can have omega-3 rich eggs, etc. So that's that. This is how the animal looks like. And yeah, I think this is it. Thank you.
Thank you. Thank you. Thank you, Pinar, for your amazing project. And I guess I'll just throw a first question, but then we'll have time for your questions if you feel like asking anything to our speakers here. And I have a general, let's say a common question, which is related to what's at stake here, which is the border between bodies and the digital. So the question is there a border? And if there is a border, is it something that somebody else decides or something that we can negotiate? And this is important because, I mean, the three of them talked about these in a way. So maybe I'm simplifying this here, but Rüdiger, an increased digitalization could make medicine and science more productive. Or Salvatore, it could make it more human, maybe. Or in your case, what's the problem with surveillance and creepy stuff that happen because our health system is increasingly privatized, for example? So this is the question. Just a quick round of answers, and then in the meantime, I can take, if you want to raise your hands, I'll take a few questions from the public.
So, did I get you right that you wanted to get a clear answer on what the digitalization means concerning biology? Well, I thought about that, and it's a very speculative idea, so it's, I'm not saying this so much as a professional scientist, it's more like a science fiction fan, is that I think the computer technology, when it develops further, maybe to quantum computing in the next 20, 30 years, I heard that that's the development time that it needs, then we will be able to simulate biology even better and simulate evolution, and maybe be even able to use the genetic codes, the genome sequences that I've been talking about, to do this translation from this abstract string of information to an idea of the three-dimensional structure, not in physical world, but in an abstract world in the computer. So that would mean that we can simulate the evolution, maybe even faster than it actually happens in real time. I don't know if it's possible, maybe. And then we can use all this extra energy that we can generate, like fossil fuels and solar energy and so on, and directly feed it into a fast-forward system of evolution, which is simulated in supercomputers. And then we can couple that back to the actual evolution. So I think the future, maybe in 100, 200 years or so, we'll see a merging of the biological sphere with the informatics sphere, which will be catalyzed by our knowledge about it and external energy sources. That's very speculative, but yeah. Stanislav Lem was also writing about it. I can really recommend his books. Thus Spake Golem. It's really cool. Like he was talking about this. He actually talked about the NSA building supercomputers, not mentioning the NSA name, but the American Intelligence Agency building supercomputers. But these supercomputers became philosophers and stopped working for the military. So quite positive vision. I hope we get these computers to use to work on evolution. I think that's a much cooler task for them.
I have a wow. I have a, it strikes me that you talk about in terms of boundaries, because the idea of boundaries is a dangerous one, no, and it's also a very delicate one. If from my point of view, it's not a matter of boundaries. It is a matter, let's say, complexity, or from my point of view, since it is a matter of complexity on a multi-level, so complexity not in terms of brute force, okay, brute force calculation, processing power, possibility for analysis, etc., etc., but multi-level qualities, okay, so the possibility to move from one level of complexity to the other. I would say that it is very interesting not to think about in terms of boundaries, but in terms of connections, because from my point of view, the most interesting and also frighteningly interesting thing that we are doing as human beings now is that we are encoding everything. We are encoding everything, so you can see it in science, you can see it in medicine, you can see it in the daily lives. What's the most interesting thing that happens for children? Children are not do not spend their time on their own anymore. They're always supervised. They're always this is the place to have fun. This is the place to. So we're progressively encoding everything, and in a sense, digitalization encodes everything even more. Okay, so this becomes interesting and also very useful if we manage not to lose the sense of complexity. So if we manage to interconnect these great opportunities of digitalization, which is very, very useful and very, but to not lose track of all the other aspects which are not related to efficiency, which are not related to brute force, which are not related to power, which are not related to anything. So encoding and decoding at the same time. So okay, fine, the encoding is very useful, but let's not forget about decoding. So connections more than boundaries.
Sure. So surveillance?
No, it's a suggestion. Maybe you have other ideas that are more interesting.
Well, as far as I understand that, like, in terms of, surveilling your thoughts or your affects, it's very costly unless you want to tweet about it every single second. And I don't know. I have this kind of dark vision. Although these companies, corporations believe that, gathering this data will, help them save money, etc., or serve better because that's their motto, etc. I don't think we'll ever get there. There are other issues that, the planet is facing right now. So they're talking about these time frames and like budgets, etc. I don't know if we'll ever get there, honestly. So yeah, maybe in like very small communities, but that's why I'm not really bothered by it. And I'm also like personally, I like monitoring what I eat, monitoring how I sleep, etc. So I don't really have a personal problem with that. But I don't like the fact that, especially again, I was born in Turkey where the health insurance system or health system is completely different than the States. In States, if you don't have health insurance, if you don't pay for it, you're screwed. And if you don't have a good health insurance company, you're even more screwed, because then you won't be able to like, go to the hospital to get a treatment. So in the case of that, like being monitored, your health being monitored this way becomes really tricky, but I don't know if they're gonna get there either. As a note, I just think that your P2P healthcare system should become really prevalent. I see a lot of future in that. And I think it's like, that's where we should go. Yeah.
Okay, thanks. So we have time for a few questions. So I have one there. Oh, there it is. And if there is more, just let me.
Yeah, something that struck me that I would think cut across all your personal experience in your fields of expertise would be how medical risk kind of operates on people, like epidemiological risk basis will like determine what treatment you get, and I think that probably feeds into the research in neuroscience and biotechnology. I think maybe in the spirit of the first Hashes to Ashes talk last night, do all of you, can you like point to some artifact that's kind of hidden from view or a bit recessive that actually does bear down on us as data?
What's the start?
I totally agree. In the sense that we are really highlighting each from our point of view a thing which is a feature which is hidden somewhere. From my personal point of view, this feature which is hidden sits somewhere across the idea of accessibility and the idea of freedom. Because obviously accessibility is a concept which is very different from the one of freedom. Some call it autodetermination, some call it autonomy, some call it in many other ways, self-representation, expression, okay? So the idea of accessibility is really, really different from the idea of freedom in all these senses. And in this sense, what I feel I'm uncovering through La Cura, for example, is this exact difference. This exact difference between the idea of accessibility and the idea of freedom, which is, if you think about it, the idea of desire, because accessibility is obviously necessary for freedom. But without desire, there is no freedom. There is only accessibility. So this, from my point of view, is the thing which is uncovered through La Cura. I don't know what you other things about.
Okay, so we even have a book in Italian titled "Open is not free."
Yes,
is that right?
Absolutely.
It's an old debate that's still lively and interesting to be discussing. So okay, so
yeah. So let's pretend the sci-fi scenarios will go in the Google's Kurzweil's direction. we'll know, and I hope we all love it, Edith. But let's pretend it goes in that direction. How can has people, you know, I might be a scientist or journalist, maybe you're more entitled to join the discussion, but how does the average person can join the discussion about the privatization of the body and actually deviate that kind of scenario?
Tough one.
If I can introduce this, it's interesting because this question relates to their skills. So of course they have peculiar skills at the very least. So hackers, designers, biologists, artists. So how do you play with that, and what do you think about what us the people can do?
If I have a quick answer, obviously, which is performance. The quick answer is performance. We as human beings, other than being encoded more progressively the whole time, have also are also forgetting the methodological indisciplinity. So we are too disciplined. We are too disciplined. We are forgetting how to, find things in the environment, how to, break the code systematically. Desertot talked about the tactics. Tactics are dying. Strategies are winning. And this methodological indisciplinity is something that could really be interesting to explore, okay? And it happens through performance. It happens through performance. So with the idea that the future doesn't actually exist, but it's a performance which we enact with each decision we make, this is, for me, of course, is an interesting point of view in starting to, okay, where do I begin from? Okay, so performance.
I kind of agree. But from practically speaking, I think that's more challenging. But if you look at the IT development in the last 20 years, 10-year-old children can learn programming, and they teach it themselves just from free protocols in the internet. And I think it's not very unrealistic that the same will happen in biological terms. So the data is open to a good extent still. It depends on what we do with the stuff that comes up now. But if we have it open, and if we get a better understanding, and if the computers get better in simulating the stuff, and presenting us how it works, so we can directly grab this sequence. Okay, this looks cool. This does this, and you just pick it and you combine it with this, and then the simulation is good enough that it actually will also work in reality and not just in a simulation. I can imagine that 10-year-old will program life as they please, and then it's also a performance. It's a real life performance. So the thoughts, the dreams just materialize in your hands by just making decisions about it.
Well, I have problems with what you said, but back to your question. So you're asking how lay people can contribute to this discourse. Well, there are institutions that claim to build the future, such as MIT, future is built there. So maybe they can go there. That could be one option, right? Or my question to you is, do lay people care about contributing to this discourse? And I believe that if they care, following some of your ideas, it's possible. I love Freeman Dyson, and he has this article called Our Biotech Future, where he believes that following the computing revolution, or a personal computing revolution, this biotech revolution will happen as well, and playing with genome will become like a common thing. And he actually says it will become an art form, like sculpture or painting.
That's what I mean.
Yeah, but I have, maybe I misunderstood you, but yeah.
Okay, thanks. We're running out of time, so we have one. I'm afraid it's the last question.
Well, it's wow, that's loud. It's a bit of a statement as well as a question, because it's you actually touched on what I was going to mention in this last question answer, and that is that now a lot of these things, what we're seeing now is the beginning of new art forms that come from big data. That's the key to big data. It's not really having more simulations or better simulations. It's that the people create their own simulations to take advantage of a lot of the big data so that you can create new art forms that answer questions that haven't even been answered yet, that can't be answered because we don't understand how to interpret the data. You see, a lot of these, like for instance, a lot of your neuroscience research, you call it art, but it's also research. You know, things like this chicken with no legs and brains. Don't be surprised if you see that in reality. Let's say that if we thought about it coming. Because I want the copyright. Yeah, I mean, but imagine. See, the thing is that you see it as an art piece, and you've gone to great lengths to create a lot of detail, and let's say that that could be possible in 10 years. Well, with big data and the way that we can interpret it, now that you've given this artistic interpretation, you could probably see that in the next 2. Technically, you probably could see it in the next year, really, if they put money into it. So it's, you're all kind of different points of view on the same thing. You're all, even though you say you're a designer, you say you're a researcher, and you say you're an artist, you're actually all kind of doing the same thing. You're all interpreting this data, you're all interacting with this network, and you're all getting results which fold back in to create new data. And then other people will come along, and I think the thing that's going to have to happen is that data is going to have to become the paint of a new art form. I guess that wasn't a question at all. I was just really, I loved your talk. Thanks. Bye.
Yeah, I totally agree with what you say. There's a book from Hermann Hesse. We get a Nobel Prize for it. It's called the Glasperlenspiel, glass bead game, and he's describing the perfect, ideal art, science, whatever life expression that you could imagine. And I think that's where we're getting now. So, and I have this vision of transforming these skill sets and these tools and these approaches into something like Hermann Hesse was envisioning. And I think we're not far away. And the way that you described with this decentralized use of data, so you make your own tools, you make your own simulations, that's probably the way to go. That's the most efficient way. So it's a bit like a SETI at home thing, but not you loading data sets and processing it, but you interacting yourself. You create your own data sets and interact it and put it online so it can be picked up by others.
Can I show you something real quick? Just briefly, just to kind of send it home a little bit. This is something I designed basically from the internet. It uses transcranial direct current stimulation. It uses Arduinos, and it also uses 3D printing to create something that creates a form of music that's based on creating neuromodulation. 3 years ago, I didn't know any of this stuff. I didn't know any of it even existed, and it's all internet. And you get to learn more stuff as you go along. So it's interesting because to see how you're all interpreting data as in your own ways, looking at your own cancer, you know, making Frankenstein bread and chickens with no legs, how all of these things are feeding into people who are in this audience right now and how they'll be interpreting this stuff over the next, who knows, maybe next week, maybe over the next year. You're part of that kind of thing that's happening right now. It's really interesting.
Thank you. So do you have any other questions? I think we really are running out of time. But I want to thank you for opening like this broad picture that we pretty much needed at this point. And Tatiana, yes.
Logistic information that tomorrow there will be a panel again with Pinar Yoldas at 12:30 related to her exhibition at the Sharing Stiftung, an ecosystem of access. So I think also we can go on with the discussion tomorrow from 12:30 in the auditorium.
Yes.
But now I leave to you to conclude.
I just wanted to ask you to join me in thanking our speakers, and it's Pinar, Rüdiger, and Salvatore, and Tatiana, of course, for inviting us. Enjoy the rest of the festival, and thank you, Dankeschön, grazie.
Woo.